There's a familiar theme today that we need to understand what happened so that going forward we'll be better prepared. And this last panel is called COVID-19, its origins and the regulation of virology. We all witnessed the toll that COVID took on the global community, and going forward, we will want to be sure that we're not solving for the wrong problem. This panel will be moderated by Jan Jekielek, and Mr. Jekielek is senior editor of the Epoch Times and host of American Thought Leaders, where he engages in deep conversations that challenge grand narratives. Thank you.
Well, it's an incredible honor to be able to moderate this extremely distinguished panel on a very, well, what's been in our country and I think around the world, a very contentious issue, right? What is the origin of COVID-19? But then there's also this other piece, which is, how do we need to regulate virology in general? And they're actually, these are two parts. So the way that the question of this panel is being posed, if it is indeed a zoonotic origin, if it is indeed a natural origin, maybe we need to look at how to minimize these contacts of wildlife with human beings, right? Or if it's a lab origin, well, then we really need to regulate the sort of the risky types of experimentation, what's often called gain of function. I guess the question I want to pose is the premise correct in the first place, right? Are both these things something that we need to do, or are both these things something that we don't need to do, irrespective of the answer? And so we're going to start. I'm going to do a super brief introduction of each panel member here before they go into their 5-minute introduction, because I think I could spend the whole panel basically explaining all their achievements throughout their careers. So let's start with, Alex Washburne. Alex Washburne is the scientific consultant at Silva Analytics, LLC. And also, if I may editorialize a tiny bit, has a vast depth of knowledge on this issue of origins, has been thinking about it since the very beginning. Please.
Thank you, Jan, and thank you, Jay and Sanford, for having us all here. I have a checkered past. I'm a mathematician and a biologist. And they like to say that the problem with being a mathematical biologist is the mathematicians think you've gone soft and the biologists think you've gone crazy. Yes. So before, I've done a bunch of things, but I think most relevant to this panel is in 2017, I was working on a DARPA grant to study pathogen spillover from bats to people. And my work was specifically focused on outbreak forecasting and viral origins. So before it was cool, I was looking into the origins of filoviruses and forecasts of bat Henipavirus outbreaks and things like that. And then during the pandemic, got involved in a lot of the other kind of contentious topics of outbreak forecasting, public health policy, helped a hedge fund short the market in February of 2020, and other kind of oddities and things like that. But at the end of all that odyssey of outbreak forecasting and stuff like that, I got back to the questions of where did this virus come from, and drew on this work from a DARPA preempt team that I was working with on the methods we use for forecasting the likelihood of a jump-capable quasi-species in the language of that call. And I found out that the diffuse grant is one of the main pieces of the lab origin theory, and it traces back to the same call that I was working on back in 2018, 2019, 2020. I got diving into the details and thinking about the methodologies of the wildlife virology, and we had this big operation catching bats in Africa and Asia and Australia and shipping samples across borders and then processing them in the lab. And from that lens, I entered the origins question and looked into whether or not the virus has any evidence consistent with the methodologies from that time. And so I've helped produce one paper on the virus looking like it may have a pattern consistent with the methods we would use to resurrect these viruses from field samples, or what's called infectious clone technology. It was corroborated somewhat in a January 2024 FOIA of the diffuse grant, which found the specific enzyme we had hypothesized could have been used for a reverse genetic system in SARS-2. That specific enzyme was in an order form for the diffuse grant itself. And so it's I think the actual considering of all the different evidence, in addition to what we worked on, but the furin cleavage site, the site of origin, the biogeography, and more, takes a lot of math and bio to really fully consider it, and I hope that we can do it justice here. And so thank you so much. I'm really excited to chat about it.
All right. Well, next we have Dr. Simon Wain-Hobson, Professor Emeritus at the Pasteur Institute, and you may be familiar with him from some of his work around HIV.
Yeah, so I'm a sort of a wet virologist, and I've been battling this, what is known as the gain-of-function risky virus research now for 12 years. I was about the only virologist addressing this back in 2012. Basically, this is taking viruses mainly from animals and deliberately adapting it to humans. Deliberately, not perhaps, deliberately. And basically, they're making the world a more dangerous place. But the shock is that these experiments cannot deliver a single result for any health official. They cannot deliver, and they have failed to deliver, and in 12 years, you will not find one good justification of why this work should be done, but it's gone forward. The scientists do not see, there's another name for GOF, it's dual-use research of concern. The scientists do not see the D, the dual use. They say, we're there to help society. They do not understand the D. The D is for the State Department, which is the lead department in Biological Weapons Convention at Geneva. They do not see the D, so they're totally schizophrenic in this, and there's been very little public discussion, good discussion about two good conferences in all. How is this possible? Bench scientists no longer have the time to care. They're so busy running after money, doing experiments in a very competitive environment, which is good. They are quick. Ethics is for other people, ethicists, therefore it's not for them. And they use words badly and very imprecisely. So this is a dangerous situation. What can be done? And I'll go from the easy to the most difficult. We need, in times of crisis, virologists are best doing their virology, but there are times when it impacts society. We need to share a little more of what we know. The public want a little more. They don't want everything, they want a little more. That's relatively easy. My wife is a pediatrician, my son is an interventional cardiologist. They've taken a Hippocratic oath. They could do harm, but as a virologist who's thought about this, I could conjure up some really nasty stuff that you really wouldn't want to know about. And no Hippocratic oath. So I think a Hippocratic oath for life science PhDs, pretty easy, fairly easy to do. Stanford could start it tomorrow if they wanted, I'd imagine. So the next thing, we need independent oversight. And Bryce will talk about this, and therefore I won't take his time. And it has to be independent of the life scientist. Why? There's a study, a two-page article in Nature 2013, showing that scientists overestimate the benefits of their work, and they underestimate the risk. This is just a fact. It's documented. It's rather nice because we think we're doing good, and we sort of say, well, there's a bit of risk. But it shows that there's rather nice people in some ways. And the hardest of all is we have to delete the adjectives, promote more humility, and endless pedagogy with the public who are so thirsty about infectious diseases and virology. And we just need to help them and explain what we're doing. And I'm sure that will take down the temperature.
Okay, wonderful. Next, we have Dr. Bryce Nickels, Professor of Genetics, the Lab Director at the Wakeman Institute of Microbiology at Rutgers University, and the co-founder of Biosafety Now.
Okay, thank you very much. So I really am here in my capacity as one of the founders of Biosafety Now. What Biosafety Now is a nonprofit that was founded we launched in February of 2023, and I'm one of 5 members of Biosafety Now that's here. Laura was one of the original members. Simon has recently joined, Jay Bhattacharya has recently joined, and Andrew Neumer has recently joined. We have a clear mission, and I would recommend for people that are trying to do anything, I recommend making a mission statement because it makes it very easy to figure out what you should do. So I'm driven by a clear mission. One is to get effective oversight. Now, I'll talk in a second about what effective oversight might be. Two is accountability for wrongdoing that has happened. But I think, and many people in this room think, that the COVID pandemic was caused by reckless research and a lab accident. And what I want to focus on really in the next few minutes is the third one, which is to give the public a voice, which is, I think, the most important thing, which is to elevate the voice of the public in these discussions, because to date, they haven't had a voice. Simon just said something really important, which is this idea of independent oversight. I'd like to just do a poll of people here, just show of hands. Do you think if there was a virus that killed, whatever, how many people were killed by the 1918 flu?
50 to 100 million.
50 to 100 million. And that virus was eliminated from the earth. Do you think that it should be brought back to the earth? Well, that's the left. Does anybody in the room think it should be?
Yes,
That's
I
fine.
do,
So
and
you
I-
do, okay. Now, that's fine. So you think it should. But
Explain why.
if you have the ability to do it, should you be allowed to do it without anyone weighing in on that? And that's really what this independent oversight is. So let's just say, should experiments like that, which present a potential for an existential risk to humanity, be done on the whim of a scientist or not? Do you think, just by show of hands, do you think there should be an independent panel that reviews those decisions? Just, if you do, raise your hand. No pressure, no pressure. No pressure. I mean, there was kind of pressure. There's honestly is a little bit of pressure, because I mean, come on. Really, you think the science? So, okay, well, keep your hand up. Keep your hand up. Because what I'm going to ask you to do, those with your hands up, you can actually help right now. This is a call to action, frankly. I'm blessed to be on the stage right now, because right now there is a bill that's making its way through the United States Senate to actually implement that independent research review board. It's called the Risky Research Review Act. And what you can do is you can support that bill. Many of you in this room, many of you are pretty famous, right? I mean, legends, I would say some of you, not all. I mean, you are, of course, a legend.
No.
But the point is, we don't need many names. So some of your names will be extremely valuable. So I'm going to email you, well, if Jay allows me to, and I'm going to ask you to support this bill. And your name will matter. Your name will be on a letter that will go to every senator that says this is essential. We need this piece of legislation to prevent the next pandemic. Because that piece of legislation would have stopped the research that occurred in Wuhan. It would have. So whether or not that research caused the pandemic or not, this panel would have stopped it. This bill would have stopped it. It's a safeguard. It's the firehouse that's being built after the fire. Frankly, to quote, I think it was Doug Owens said, we shouldn't be building, we don't want to build, what do you say? Don't wait until you have a fire to build a fire department. Well, we've already had a fire. Let's build a fire department before the next one. It's a simple thing. It's a simple thing. Independent oversight, about 60% of you raised your hand. The others, I don't know. I'll come over and talk to you about it. But anyway, thank you. It's something you can actually do right here, because panels, we can talk about problems, but you can actually help on this one. This is no joke. This is no joke. And Jay will confirm this. Jay, do we have the ability, if a few people in this room put their name on this, will it move? Fast track in the next 2 months. I swear. We can do it. You just have to step up.
Well, I'm going to write an email to say the contrary if you do that. So
That's fine.
we'll have
That's
to
fine.
see what actually
Well, an email that, so I guess I'll stop so we can go. Yeah.
That's right. All right, well, Dr. Laura Kahn, co-founder of the One Health Initiative, and it would also be good to understand, I'm sure you're going to tell us a little more about what that means.
Right, thank you. First, I'd like to thank Jay and the organizers here at Stanford University for organizing this really important conference, and thank all of you to sit through all of this until our panel. So thank you for your attention. Well, first, before I start talking about One Health, I think it's important to determine what exactly is the evidence needed to prove the origin of the virus. And if one wants to conclude that this was a natural spillover event, then we need to look at what was the evidence that was used for SARS-CoV-1 back in 2002, 2003, and what was used for MERS back in 2012, the evidence that was accepted that these were natural spillover events. Now, so for SARS-CoV-1, and I apologize I can't share my PowerPoint slide with you, but the evidence for SARS-CoV-1 was that there was animal antibody and viral genome evidence from palm civets in the animal market in Guangdong, China, had genome sequences that were 99.8% identical to SARS-CoV-1. 80% of the palm civets tested positive for the antibodies. In addition to that animal evidence, there was also occupational serological evidence. A study of 800 people in the Guangdong province found that people who traded in masked palm civets had the highest positivity rates, 73% of IgG antibodies to SARS-CoV-1. Looking at MERS in 2012, the animal antibody evidence was dromedary camels throughout the Middle East and parts of Asia tested positive for MERS-CoV antibodies. And in fact, archived camel's blood specimens dating back decades tested positive for MERS-CoV antibodies. In addition to the animal data, there was also occupational antibody evidence in the workers. A study of over 10,000 adults from all 13 provinces of Saudi Arabia found MERS-CoV antibodies in 0.15% of the samples. In camel shepherds, antibody seropositivity rates were 15 times higher, and for slaughterhouse workers, they were 23 times higher than there were in the general population. So that data for both SARS-CoV-1 and for MERS-CoV were the acceptable evidence to determine a natural spillover event in those outbreaks. Now, when we look at COVID, was there similar data? And in fact, there was not. Neither the virus nor antibodies to the virus have been identified in animals in Wuhan during the early period of the epidemic. In fact, there was 0 out of 457 samples taken from 18 species of animals sampled in the Wuhan market tested positive for SARS-CoV-2. In addition, there's no correlation has been observed between human occupational exposures to the animals and higher rates of infection or seropositivity to the virus. And the Chinese had had experience with SARS-CoV-1, so they knew what they were doing. By early 2020, Chinese physicians had conducted serologic surveys of thousands of people to assess the prevalence rates of SARS-CoV-2 of antibodies. Unfortunately, none of these studies included occupational data. Now, whether they had done occupational data and did not report it, or it was simply not done, which is surprising because they did do it for SARS-CoV-1, we don't know. Nor is there any data on the laboratory workers. So we simply do not know. Now, the expectation of that data coming forth is up for debate. But if this were indeed, so there's no evidence that there was an animal market spillover. If we expect the Chinese to come forth with evidence saying, oh yes, there was a laboratory spillover, I think it's unrealistic that that's ever going to happen because they have zero incentive to do that. So if that's the criteria needed for a laboratory spillover, then we are never going to have it. And I'll be happy to elaborate more on that in the Q&A section. Thank you.
Okay, great. And we can talk a little bit more about One Health after. One thing, just for the benefit of the panel, I want to start, before I introduce Dr. Gupta, I'm going to start with talking about the origins question, maybe for the first part of the panel, and the second part will be on the regulation of virology and so forth, and the Risky Review Act and so forth. So without further ado, Dr. Sunetra Gupta, Professor of Theoretical Epidemiology at the University of Oxford.
Thank you very much. Okay, what I'm going to do, I thought, especially since I'm quite jet-lagged and it's late, and this is my second panel, is I'm going to just read out what I wrote for Prospect magazine when they asked me to make a small comment about this whole theory. Someone had written an article, and this was supposed to be a reply, but it was solicited. So this is what I said. I said, to my mind, the discussion about the origin of SARS-CoV-2 rotates around two equally implausible scenarios. Either that the virus was manufactured in a laboratory in Wuhan, or that it jumped out of a dish of pangolin chop suey somewhere nearby. What is far more likely is that the virus had already been circulating over a period of several months in China and only came to light in Wuhan. Just because we found it in Wuhan doesn't mean it came from Wuhan. So it only came to light in Wuhan when it was at sufficient prevalence for an intelligent physician to identify an atypical cluster of hospital cases and raise the alarm. I cannot blame my colleagues for rushing to stem potentially dangerous speculations about a leak that might seem plausible from the coincidence that the virus was first detected in such close proximity to the Wuhan lab. However, it is extremely unfortunate that a civilized discussion about the possible origins of SARS-CoV-2 became impossible under the dedication to a single narrative that was subsequently supported and sponsored by them, not just regarding the origins of SARS-CoV-2, but also with respect to the infinitely more important question of how to come up with a humanitarian solution to the problem of minimizing deaths from the pandemic. I'm going to jump into the gain of function. I should also like to make the point that the hysteria around the gain of function, is misplaced. Sorry to be not entirely respectful of that word. Those who are clamoring for it to be restricted would do well to recognize that many of the proponents of lockdown were also firmly in favor of stopping gain of function research in pre-pandemic times. My own assessment of gain of function, which I believe is shared by many other virologists, is that it's an unfortunately named standard process for determining the properties of viruses and carries a very low risk to any of us. I can substantiate on that. In fact, gain of function experiments are far more likely to lead to medical interventions that improve the lives of people than to spawn a new pandemic. Nature is far more adept at that. So what I'd like to do is actually go back to the framework that I used in the last panel to think about lockdowns, or the global solution to pandemic threats. A problem of pandemics, which I broke down to three things. The threat of it, can we actually do something about it, and what is the collateral damage? So the threat of gain of function, the threat that arrived, well, this is actually more all about gain of function. Do you want me to keep this to the second part? The talk?
This is the perfect foray into the
Okay.
first part, which will be about the origins question, right? Because the premise is that gain of function was used to create the virus.
Yes, but I've just suddenly realized that this part of it is more about just assessing our response to gain of function research itself. So perhaps what I should do, I'll save that for later and just respond to because I have been vocal while Bryce was speaking and also Laura. Just say why I think I don't buy these arguments. So with regard, this comment about 1918, the flu virus of 1918, that it would kill everybody. We actually have evidence that it would not. In 2009, we had a pandemic, swine flu pandemic, which was, at least in terms of its hemagglutinin protein, almost identical to the 1918 pandemic. Some of the work I've done replaces actually the standard theory of how flu evolves, which is a sort of linear narrative, with this idea that it's more cyclical. And indeed, the 2009 flu pandemic proved that. It was basically the 1918 pandemic strain coming back. It didn't kill 50 million people because flu now is a seasonal pathogen. It wasn't in 1918. It was pandemics would come and then they'd go, and you'd get 30 years with no flu, and then it would come back again. But since 1918, it's become a seasonal phenomenon, and effectively we have enough immunity now to protect us from severe disease, even when a new strain arrives. So the idea that the 1918 flu virus, if reconstructed, would cause this massive new set of deaths, I think, is incorrect. I think that it is wrong to go after Taubenberger and people who did do that work, because I think all it's brought to most of us is the benefit of knowing what the 1918 virus looked like, particularly from an antigenic point of view. And in my own work, I'm currently trying to make a universal flu vaccine. It's actually provided us with the clue of what to put in that vaccine to stop it to use it to stop people dying of that same strain when it comes back again. With regard to not being able to find it in animals, well, the fact is that clearly it's a specific evolutionary event which may or may not make it transmissible in animals. And in the case of SARS-1, it happened to be something that was around in other animals. That doesn't have to be the case for SARS-CoV-2. SARS-CoV-2 may easily have just been spreading, may only be something, it seems to be, in fact, clearly, that is specifically adapted to humans. So it could have been spreading through humans in China. How do we know? What do we know? We have no evidence that it wasn't doing that. So I think it's more likely that it was. MERS is actually an endemic infection in many parts of the world. You bleed the Turkana tribe, 50% of them have antibodies to MERS. These are different types of viruses. They have different epidemiologies. SARS-CoV-2, I think, is very likely to have emerged from some bat or something somewhere else in China, been spreading through humans, got to Wuhan, was detected there. That, to me, is the most parsimonious explanation for where SARS-CoV-2 came from.
So Dr. Gupta just jumped into the first question, which is, we ran it a little longer. This is, so you're providing you're thinking about, the evidence that natural origin is the more likely scenario. Let's, from our other panelists, who I think are side towards the lab leak side, let's just kind of enumerate the evidence, in particular, I guess some of the specific, like for example, Alex, you found these very specific restriction enzyme fingerprints, right? This is a very interesting piece of evidence that's valuable to consider. Let's go through the remaining panelists and just set up the evidence for lab origin.
Absolutely, yeah. So first I want to let everyone know that there are a few people in the world I admire as much as Dr. Gupta, and so I'm really excited to hear her thoughts on this. And from the lab origin theory's perspective, it really boils down to probabilities. And even though there are two equally likely scenarios, the likelihood ratios may not be similar, at least when we try to do due diligence and use prior methods to estimate those likelihoods. So one example is, what's the odds of this endemic, let's say, coronavirus emerging in Wuhan of all places? SARS coronaviruses span this region of Southeast Asia that contains about 1 billion people. Wuhan has about 10 million people. So just off the cuff, you might estimate if you were sitting in my chair in 2018 trying to forecast the emergence of bat viruses, you'd estimate a 1% chance that Wuhan is the site of origin of a SARS coronavirus. The other thing to look at is SARS-CoV-2 has this interesting motif called the furin cleavage site that in 2018 and before, people were studying the roles of these proteolytic cleavage sites that the virus cuts open the protein and uses that to then engulf the cell, cuts open the viral protein, engulfs the cell. So we knew some of these mechanisms by which these proteolytic cleavage sites enabled cell entry. And we were thinking about these as well as like what that could be a mechanism for jump capability. But when you look at the evolutionary tree of SARS coronaviruses, there's about 1,000 years of evolutionary time. Now, granted, it's not equally sampled, and we haven't been going back 1,000 years, but we can reconstruct a tree, and the branch length is 1,000 years. And there's not a single furin cleavage site within the SARS coronavirus clade until a year and a half after people proposed to insert one. Where did they propose to insert it? In Wuhan. So now we have a Wuhan origin, we have a grant proposing to insert the strange motif in a SARS coronavirus in Wuhan. But then how would they do that? In order to insert a furin cleavage site in this RNA virus, the RNA virus has a single-stranded RNA genome that's really wobbly, and you can't actually just insert things into it. You have to build a DNA copy of the virus. And it's not only just to do the insertion, but the protocol for getting bat samples out of Laos and shipping them across borders to then study the virus in the lab required typically inactivating that bat virus sample so that it didn't infect people along the way. And then you have an inactive virus in your lab, wherever that is, and in order to reconstruct it and study it, you have to build this DNA copy. So we looked at the viral genome of SARS-2 to see if there was any evidence consistent with the method called infectious clone technology or reverse genetic systems that we use to build these DNA copies. So you typically print out chunks of the virus, and then you glue them together with these cutting and pasting sites. So the pattern of these cutting and pasting sites was unusually evenly distributed, consistent with that prior methodology. We estimated the odds of that were 1 in 100 to 1 in 1,400. But then it goes even further. If you look at the mutations that bioengineers use to move around these cutting and pasting sites, they exclusively use silent mutations. So every one of these cutting and pasting sites that's modified in SARS-2 relative to its closest relatives is moved around with silent mutations, and there's a much higher concentration of these mutations within these cutting and pasting sites than the rest of the genome. So if these are just random mutations happening throughout the genome, they should be equally spaced and not concentrated within these unusually spaced cutting and pasting sites. Our estimated odds of that range from 1 in 20 million to 1 in 50 billion. So being the statistician behind that and kind of unveiling the curtain of these statistical tests, that was the point when I started speaking up a little bit more about my estimates of the odds of a lab origin. But I think it's still worth considering alternative scenarios so we don't have blind spots, this possibility of endemic transmission or things that we're not considering when we focus on a wet market, like what if a cat ate a bat? This particular furin cleavage site found in SARS-2 is also found in a feline coronavirus called FIPV. Now, that's where these two theories don't really discriminate against each other too much because on one hand, okay, maybe a cat ate a bat, and maybe a cat had another coronavirus infection and a recombination event happened. There's all sorts of possibilities in evolutionary biology. But one of the authors of the grant proposing to make this reverse genetic system and insert a furin cleavage site in a bat SARS coronavirus in Wuhan also mentioned in congressional testimony that they had these feline coronaviruses in their minds when they were thinking about what sort of furin cleavage sites to insert. So this is the kind of uncomfortable situation of being a lab origin theorist is some of these people I had worked with before COVID. And I went to EcoHealth's headquarters in 2019, I published a paper with some of them in 2018, and now we're disemboweling their grants and studying this similarity between their grants and a coronavirus, and it's very uncomfortable, but that's the sort of thinking we have to consider with a lab origin theory is very focused studies of the methodologies used. And one thing I haven't said yet is that this particular cutting and pasting site pattern in SARS-2 with the high concentration of silent mutations is with enzymes. These two enzymes have only been used together on a coronavirus infectious clone once before, pre-COVID, and that was by this gentleman Ben Hu and colleagues at the Wuhan Institute of Virology. And so there's some kind of ways in which there's a consilient lab origin theory revolving around this grant. That doesn't mean everyone who wrote it acted on it, but with that grant circulating, if you gave me that grant in 2018, and I used that to predict the emergence of SARS-2, it would be much a more accurate prediction than using the methods of the time for wildlife, biogeography, and viral emergence.
Can I jump in on that diffuse proposal for a second to say it's very important that we discuss where the research was actually proposed to do, because the two scenarios have been laid out as research in Wuhan or natural. However, the research proposed to insert furin cleavage site included Ralph Barrick at UNC, and in fact, the work was proposed to be done at UNC, and in fact, my understanding, I believe, is that we don't have the full grant proposal. It hasn't been released, do we? Correct? The US government has not released the full grant proposal, which would include details that might help us know where that research was actually really proposed to have been done. We are basing the idea that it was done in Wuhan because there's a footnote in a draft that says we can offload, well, I'm paraphrasing it, we can do this research in less biosafety levels in Wuhan because it'll, we don't want to, I forget exactly. Let's get our facts straight about this. We don't know if the research was literally proposed to be done in Wuhan or not, correct? That is an assumption. We would need the full diffuse proposal to see that. And I would actually, if this was live streamed, this would be something, again, all of you can help. What we need is the documents released to the public. Get the public involved. All of these investigations, these committees, they deliberately release a select subset of documents so they can control the messaging. They should be releasing all of the documents, frankly. And we can all get that to be done. But so the directing all of this to China, I get it. But frankly, there are people in the US, Ralph Barrick in particular, who has to be answering more questions about his role in this research. So apologies for interrupting, but it is a source of frustration for me because the guy is, giving a keynote in Japan in a few weeks, right? Like he's still a star of the virology community. The virology community has let everybody down. And we can talk about the cover-up. I think that's what we really should be talking about because the cover-up is happening actively now. There are documents we don't know. We have limited information, and it's because this is more than science. It's about some geopolitical nonsense to try to keep us out of some, I don't know what it's to try to keep us out of. But frankly, if we're talking about trust, transparency, we really, the only thing we can do right now is correct the future because there are too many people who have, I guess I'd say, blood on their hands or culpability in some level or screwed up. They don't want us to know what happened in the past. So it's frustrating. Anyway, sorry. I'm sorry, Alex.
Quick follow-up on that. Let's see, so Diffuse was not funded, and DARPA, to their lasting credit, they invented the internet and they did not fund Diffuse. Yet, there's other collaborations that were happening, there were other funding sources that were happening, and I do agree that the best way to disprove lab origin theory would be transparency. And that's been unfortunate we haven't had it.
Yep.
Lisa.
In 2004, the National Academy of Sciences issued a report, Biotechnology Research in an Age of Terrorism, and they listed 7 experiments of concern. I'm not going to read off the 7 experiments, but among those experiments included the expansion of a pathogen's host range to make a pathogen more virulent and to make a pathogen more infectious, to allow a pathogen to evade diagnostics and to make it resistant to antimicrobials. So one could argue that gain-of-function research of a pandemic potential pathogen falls under the rubric of one of these 7 experiments of concern that the National Academy of Sciences listed. Now, that's not to say that all of gain-of-function research is bad. And you can look at the insertion of insulin genes into an E. coli bacterium to produce insulin as a way to cost-effectively produce a drug that's needed for millions of people. That's gain-of-function research. The bacteria don't normally have that capability, but this research allows it. So that's beneficial. That does not involve a pandemic potential pathogen. Now, we could argue over the definition of the reconstruction of the 1918 influenza pandemic virus. Was that indeed gain-of-function research? Well, it was resurrected by that research, but if we looked at the strict criterion, they did not give the virus additional capabilities to make it more infectious or more virulent. It was the reconstruction of it. And that did yield some useful information. However, it sent horrible signals to other nations, and they interpreted that research as, oh, well, the US is reconstructing this virus, and that could be considered perhaps a bioweapon. So from a political standpoint, then, that research could be argued to be problematic, even though it yielded useful information. So sometimes this research can have unintended consequences that we might not want. So I'd also like to say that virologists have been inserting furin cleavage sites into SARS coronaviruses since 2005. So it's not a novel type of research. They wanted to know, people are curious. The scientists wanted to know, why do some coronaviruses have furin cleavage sites and why do some not? And they found that these furin cleavage sites do indeed make the virus more infectious, possibly more virulent. So this is not a new scientific inquiry. It's been done for many, many years. We've gotten better at it. And the question is, what biosafety level was the research done? And of course, we have to respect Mother Nature, that these microbes aren't entirely predictable, and accidents do happen. So whether or not an accident happened in Wuhan, it's certainly possible. The laboratory was reported to have some biosafety problems by some inspectors from the US. And so it is certainly a plausible scenario, also given the fact that there's no evidence to the contrary that it was a natural spillover event. So.
And I guess, so maybe for Dr. Wain-Hobson or anybody else, is there any other evidence sort of for lab origin that you would like to present at this point? Because the question I also want to ask is it ever possible to know with certainty the answer to this question?
If we benchmark the COVID origins on HIV, for which we've had a lot of time to work it out and there's a consensus, it might surprise many in the audience that the natural theory is woefully unproven when you benchmark on something like HIV. That's not to say it's not the case. Science needs time.
What do you mean exactly by benchmarking with HIV?
We have an agreement, or we have consensus on where and how HIV emerged from chimpanzees in Central Africa. And there's lots and lots of anecdotal data, fascinating, but not for today. And we don't have that level at all, even though we're 40 years on, and therefore we can sequence faster. We're not at that level, therefore the natural case for me is certainly not closed and tied up. And we need to give science more time, as always. There's been a lack of cooperation from the Chinese, so therefore your remark about certainty, I think in science we try not to use the word certainty. We try to use evidence and logic, and, as you know, but it is we probably will not know. That's my prediction. But I think there is information around, and more can be done. As a virologist, I'm terrified it could be the case. Because 20 years ago, and I came through HIV, and the first thing with origins was, where did it come from? Everyone said, was the KGB, and everyone said, no, it's the Americans. And the answer was, at that time, there was no one clever enough to have made such a virus that was so diabolical. So it was clearly natural. We can now make viruses, we can adapt, we can make things that you don't want to know about. And there's a paper published, funded by the NIH. They made a flu virus that was transmissible through aerosols. And what was the kill rate in ferrets? 60%. An airborne virus with a kill rate of 60%. That's in Ebola, that's lower than HIV. And this is done, and the only person, the only dog that barked was this man, this Joe. So I'm an old dog. And no one else. And they said, no. A respiratory virus with a 60, do you realize? I mean, you actually understand. You've got lots of epidemiology and public health. You know better than me what would that mean.
Well, and exactly, I was just going to go to Dr. Gupta here. So is it indeed possible to make viruses like this that will function that way? And
People forget. So you're talking about Ron Fouchier's experiments?
No, I'm not. I'm talking about Troy Sutton.
Right, okay. I was thinking Ron Fouchier's experiment. I mean, what you have to remember, or what we must remember, is in the evolutionary ecology of these systems, what's limiting the spread of a virus is not just whether it comes into existence or not. That's just an event. And these viruses, they have high mutation rates, they recombine all the time. I can assure you that they are interrogating a lot of the possibilities for what they could possibly be. Then why don't we get an explosion of new viruses all the time? It's because new viruses have to compete against the existing viruses. They have to compete on a landscape of immunity that exists towards related viruses. And when you put all of that together, you realize that a virus that you've made by passaging it through 1 million ferrets until it can actually go from one ferret to another is not necessarily, is actually very unlikely to make it in the wild. It's like you can squeeze it into a box. You can keep selecting and selecting and selecting until it does something that it doesn't normally do in the wild. But actually, that virus is not going to compete very successfully
Then why
in
do
the
the
wild.
experiment? Why do the experiment?
Because you want to learn. Why do we do experiments at all? We create artificial systems.
No, I
we
disagree.
mutate things.
I disagree.
Why?
When you write your grant, and remember, the cutoff is sometimes as low as 10, 12%. You have to give good reasons why you do it, just to sort of say, well, I want to do this experiment because I think it'll be great.
I don't think that's what any, I would think that's a...
are wanting to know why you, and it's in competition with other grants, and just to sort of say, because I want to do it, I don't think is, or because it can be done, I don't think is...
I very much doubt that that's what was written in any of these grants, that we want to do it because it can be done. Obviously, the reason why people want to do it is because it's useful to know. It's very important to know what the virulence factors are. It's very important to characterize them. And in terms of making something, a virus, a viable virus, at least in my experience, because even though I'm a theoretical epidemiologist, I actually do have a lab, it's really very difficult to make a viable virus in a lab. It's extremely difficult. It's difficult to make anything that even vaguely infects a cell. It's really, really tricky. So if I could now maybe go back to this whole idea of what gain of function, why gain of function is, I mean, how we should be thinking about gain of function. First of all, I do think gain of function, I think Laura's actually explained quite nicely, it is a most misunderstood term. It is almost as bad as the term herd immunity, which was grossly misunderstood. Clearly one performs experiments with, I mean, you have to trust the scientists that they do perform experiments in order to understand a system so that we can get the measure of it and make design interventions that will stop,
I beg to
severe
differ.
disease. But
I beg to differ because, you see, we've got to get into the specifics and not the general. The Troy Sutton experiment was done with an ostrich virus, H7N1, for which there was not, when it was done, there was not a single case of H7N1 in a human, not even one spillover.
I very much doubt that if you look at the serology of H7.
So that is that. The second thing,
It's not
it
true.
was done, published in 2014, and it came from an isolate made in 2001. That's a 13-year period, and as we know, flu isolates vary. What is the use of that, apart from saying it can be done? And I could find no justification for that. Yes, the experiment can be done, but in terms of understanding what the potential of these viruses are, was zero for myself. And if it had been H7N9, then we'd had an outbreak in China, in humans infected. You could make a case, but not for H7N1 from an ostrich.
Okay, what do you think?
I think just because an experiment can be done doesn't mean that it should be done. So, for example, the argument for doing a gain-of-function research on a pandemic potential pathogen is to be able to predict a pandemic. But by that argument, one could argue, well, we could seed clouds to be able to predict hurricanes. Deliberately seed clouds, so we'll know it when we see it. We'll know the conditions needed to create a hurricane. Now, obviously, we are not deliberately seeding clouds to create hurricanes in order to predict them. We are inadvertently seeding clouds through our greenhouse gases, and we are creating worse hurricanes. So that's inadvertent experimentation, if we will, and Hurricane Helene has shown us what that folly results in. So in terms of doing such research, there's been a long history of oversight in human subject research because of historical abuses that raised a lot of public outrage over, the World War II, the Tuskegee experiments, other experiments that were considered highly unethical, led to a whole infrastructure of institutional review boards to review the ethics of the proposed experiment. There has not been a similar mechanism for biotechnology research. And if we do decide as a society, and if there's enough public engagement to demand such research to be going forward, then we want to create a mechanism beyond just simply biosafety that was developed in the 1974 Asilomar Conference. That was here in this area headed by Stanford University to create the whole biosafety levels. I think we need a new system for ethics oversight for biotechnology research in an analogous way that we've done for human subject research. Whether or not this research was a natural spillover or whether it was a laboratory-acquired infection, that doesn't negate the need for oversight since our capabilities have expanded so much that we need to ensure that something like this doesn't happen again, certainly not from a laboratory-acquired infection, of which there's actually quite little oversight, or not enough oversight, I should say, needed for such work. So,
Can I just say something before we move on? I mean, I'd like to talk more about, but we didn't really address Alex's, the points that you addressed, which I do think that there should be, well, we all know, that there should have been a civilized debate about this. And the points you raise, I think, are very important and certainly caught my attention Because I do think it's very unusual, and I know that, there have been rebuttals from the people who think, don't buy your arguments. But the problem is that the whole atmosphere and the language and the culture around that has been very toxic, and that's deeply unfortunate. So before we move on to what actually happened and what I think of gain of function, I'd like to go back to that properly at some point. But I just want, before we get lost in those weeds, just to say that, obviously you are right to raise these concerns, and to be dismissed summarily in the way it was atrocious. I just wanted to make sure.
Yeah, I think at one point they called it kindergarten molecular biology.
No, that was
concept
just,
of
but what we mustn't do
reverse
is
genetic
confuse
system.
Christian Andersen's appalling behavior with
Yeah.
the possibility of whether it was a lab leak or not. That's all.
Absolutely, yeah, and I really appreciate these discussions that we can have, because there are no one person can see the whole elephant of life. We're all just feeling little bits and pieces of it, and this is where I think getting good-hearted people together who see things from different angles can help us advance the issue. And I really appreciate your point about human subjects research. From a perspective of kind of the finance and risk management that I did on the side, human subjects research exposes people to liability. And if you do a bad human subject study, you can be sued, whereas the liability for causing a pandemic is unbearable. And I think there's no way anyone could recover the costs if someone were to accidentally cause a pandemic, whether through negligence or malice or just some dumb accident, which happens sometimes. Mistakes are made and humans are imperfect. And I think that's one angle that I've entered into this discussion is, would it ever be possible for someone who makes a mistake, or if we create a vial that some bad actor is able to grab and repurpose, would anyone be able to recover the costs? And I think COVID has shown us that no, they couldn't. And I think that's a useful framework for...
I think it's actually, there's no international treaty that allows that. Therefore, on the technicality, you won't be able to.
So there's actually been, a bunch of published literature, and of course the most famous one being...
Hold on, with Alex's thing, we need to talk about the published... Alex's thing is really important about this published literature nonsense. Alex had a paper that should have been published in eLife. The editor violated the journal's policy, just flat out violated that policy to...
Quick update on, this saga.
And again, everyone in this room, if they cared about that, we could cause a ruckus. No one cared.
But that's a very different issue. This is how they behave. This is how science works.
Who's the they?
Well,
who's the they?
I'm not going to name names, but I had the same problems. Every time I sent a paper to Science, Caroline Ash would write back and say, I cannot publish this.
No, this is not that at all. They have a policy that says if they review his paper, it should be on...
science works right now. I think conflating this with, they must be hiding something, I think is actually
Are you familiar with this episode?
Yes, I am familiar, and this is what
No, his eLife paper.
I know what happens to these papers. I know these journals. I know these editors.
No, the eLife policy is to
how-
take
real quick.
Yeah, go ahead, please
I
explain it.
definitely agree with both of you. Even though you're disagreeing, I agree with both of you that...
I don't know if that's possible here.
Yeah,
He's very good. He's really good. He's good.
No, but
He's so good.
But no, I think
Because you can.
on one hand, the handling of our paper was atrocious, and it undermines trust in peer review or the publication, the whole concept that you can trust what's published and unpublished, or evaluate information based on whether it is or isn't published. We submitted our paper for peer review, and the editor accepted it and billed us. And then as they were gearing up to publish the paper, they fired the editor for expressing his long-held pro-Palestinian views, immediately replaced him with someone whose first action was to reject our paper on the false claims they couldn't secure unbiased reviewers. Since then, one of the reviewers has come out and actually wrote a really great peer review. And so there's obviously a lot of politics happening behind the scenes. And that's unfortunate. That is how
Peer review is rotten to the core right now.
Yeah, that
we
is
know
unfortunately
that.
the
You can't
sausage factory.
impute from that these people were covering up something that happened in Wuhan. I mean, I just don't see how you can. Not with any certainty.
I don't think I wasn't saying, I was saying part of the issue we have has been the use of the journals to drive one narrative. The bigger point,
It's
right?
disgraceful.
So, and what has not only happened, you have pressuring of editors. Of course, five of the papers that have been published that say there's a natural origin, there are calls for retraction that I've been involved in having, put out there.
I think the idea that it spilled over and, from raccoon dogs playing mahjong in the market is bonkers.
Right, and so the way in which the journals have been
Yes.
co-opted to push that narrative is
We
a
knew
major
that. There's no surprise.
No, the public didn't know that. The public is outraged.
Yeah, the public now knows a lot of things after the SARS pandemic. And one of the things they know is that peer review.
Specific to bio is the math-bio straddler here. Math and physics and other fields are not as journal obsessed. In fact, they kind of look down on biologists for just overhyping papers and publishing them in Nature and left and right, when what they should be doing is focusing on methods and rigor. And we can evaluate papers. All of us scientists could read something published or not.
As Simon just said, the whole industry, it's become this sort of competitive kind of landscape where what works is whether you can get your paper into a high-impact journal, which is nonsensical. But, these are all separate issues. Should we, on the basis of all of this, declare with certainty that this was a lab leak? I think not. Should we, on the basis of this, ban gain-of-function research? I don't want to keep going on, but I have something to say on that point, but tell me when is the appropriate.
Right. Well, so before we go into the regulation question, I absolutely would like you to say it, but let's just ask this one question of the hands. Why aren't we having this discussion at a whole suite of other conferences? This is an audience question that someone posed. I wanted to see if anyone wants to weigh in on that.
I think it's because the lab origin is just extremely uncomfortable. When you go to another conference, the virologists who work on this are in that room. And so there's this kind of invasion of this, I don't know, to point at an actual person and to say their research is key for understanding the lab origin theory of SARS-CoV-2 makes it very difficult to have this discussion, especially when that person is colleagues and colleagues with colleagues. So there is a sense in which it violates this community that the scientific community has, and I think that makes it very uncomfortable to talk about.
There's, if I may say so, there's a US dimension. In 2014, there was a conference in Hanover, which I helped organize on gain-of-function. 2 days later, there was a meeting at the National Academy of Sciences on the same subject. The German meeting, the food was superb, and the discussion was superb. It was the best meeting I've ever had. Came to the US, we had Coca-Cola and bad sandwiches, and the discussion was staccato, and there was so little of it that the discussion sessions collapsed. And the thing, you see, it was because the US funded the gain-of-function. And we had a very fluent discussion in the US. In both meetings, there was Fouché and Karoka, so the protagonists were there, and the discussion was extremely awkward here in the US. Just observational.
Okay. Dr. Kahn.
I think we need to come back to that word trust that was discussed in previous panels. The largesse of the taxpaying public to fund research is their trust in the scientists to do what's beneficial for humanity. And if that trust is breached by irresponsible or unethical experiments, then there's going to be a backlash against funding that research. Plus, I think that scientists like to think that their work is beneficial for humanity, and the thought that perhaps an experiment has gone awry might be too painful to discuss, and they just want to shut it down and not acknowledge it. But that's not the way to get public trust. We need to be transparent. We need to be honest with the public and say, maybe there was an accident. and we need to investigate it. And if there was an accident, we need to ensure that this doesn't happen again, and that our experiments are ethical and that there's oversight, because we want to benefit humanity, not to harm humanity or the other species on this planet. So I think that might be a reason why discussions have not been forthcoming.
So gain of function is something that's been very contentious for a while. In fact, it's been banned by past administrations. It's been evaded in various ways. Let's go back to this question that Dr. Gupta started discussing of what are the arguments for, and is it not as dangerous in certain contexts as some
Okay,
people are
so
saying?
yeah, exactly. So this is what I want to address using the criteria that I established in the previous panel, which is, what is the threat? I think the threat is hugely overblown. I think I've already explained why. I think that it's very difficult to make a crazy virus in the lab. I think most of these insertions of furin cleavage sites or polybasic cleavage sites are happening in nature all the time anyway. And what we have to remember is the competition is what's driving this, and immune selection is the main driver, not whether or not this thing exists at all. I have been arguing for gain-of-function studies against people like Mark Lipsitch, and indeed Tony Fauci was very nervous about gain-of-function. I think there's a certain safetyism involved here, is that, oh, gain-of-function, oh dear, what's that going to do? So I have been arguing for so-called gain-of-function studies, which I think are just standard ways of exploring the properties of viruses, which often lead to, I think, very useful insights, some of which I'm currently exploiting in making a universal flu vaccine. Okay, so I think the threat is overblown.
Question, clarification. Yeah? When was Tony Fauci questioning? Is this before 2011?
Mark and Tony were talking, and I guess, yeah, before around 2011. Mark was really worried about Ron Fauci's experiments.
But not Fauci.
And then, yeah, they were all talking to each other and saying, oh, I remember Tony Fauci being pretty worried about it.
But he published an op-ed in the Washington Post defending it.
Yeah, then maybe he changed his mind. He's not averse to changing his mind, as we know. But there was certainly a time when Mark Lipsitch remains very concerned. And I made the same arguments to him. I said, I don't think you can make something in the lab that's going to be able to compete successfully with what's out in the wild. Anyway, so I think the threat is overblown. I think then the second point was, so even if it does exist, if there is the possibility, and indeed there always will be a small possibility that at least that you could make this, can we actually prevent pandemics from arising by stopping gain-of-function studies? I mean, I think I'm worried in this whole sort of narrative about it was a lab leak and we should just stop gain-of-function studies and then I'm not saying that you're suggesting that if we stop gain-of-function studies, we won't have pandemics anymore. But there is a sense of control, again, I think, of us being able to control a process, which I think we really can't. I think pandemics are going to occur over and over again, and stopping gain-of-function, I don't think.
I think you need to first specify gain-of-function research on pandemic potential pathogens, not just on your random E. coli that you're inserting.
It's not random, that's what, yeah, okay. So
pandemic potential pathogens.
So first
of all, it's a very tiny subset that we're talking about.
Okay, so that's an interesting point, because what I was going to say next is, and you're doing that, but not everyone is the problem. So the third point is collateral damage. And I think the collateral damage is that all of this kind of research is perceived as being dangerous. Mm-hmm. Now, of course, yes, if we could, say, restrict it to things that have pandemic potential, I just don't think We know enough about what has pandemic potential.
But
I mean,
we
you've just...
cannot do the experiment. It's unfalsifiable because if you make a pathogen, you can't inject it into people, and you can't set off a pathogen and say, oh yes, that experiment makes a pandemic because people won't.
Well, so what are we supposed to do? Not make...
It's
What if...
unfalsifiable. Therefore, you can say, I think that this could have, and you cannot get better than that.
Well, I think you can get better than that because you can say, what is the likelihood that this thing will cause the next pandemic?
No, if we get into pandemic...
And in order to do that, you
predicting
need
the
to have an
strains,
evolutionary,
our track record
ecological...
on predicting pandemic strains is zero. Now,
Yes,
people
that's
pretend-
because it's not possible.
There you are. You've answered your question. Thank you.
Yes, I know, but that means that does not mean that we should stop research into important areas.
Let me just say that Mother
infectious
Nature
disease.
is very capable of killing us. We don't need to help her.
I don't think we are helping her. I
Well,
think it's
maybe we are.
hubris. I think it is hubristic to think
It
we
is
can even
hubristic.
vaguely
It's like Icarus
impact.
flying too close to the sun. Scientific
I
hubris.
think there is very little.
We're not infallible.
But the key thing, which is actually to your point, is that we literally have something in front of us right now that we can discuss, that bill, which doesn't ban it. It just subjects it to review. And I think you would...
Whose review?
Well, it's 9 people, and they're appointed by the president, and they are supposed to have expertise in life science research, national security, biosafety. They'll be vetted, they'll be
What will
recommended
they-
by the Homeland Security, Government Affairs.
What are they going to ban? If they
They're not gonna
ban,
ban
like you're
anything.
saying,
They're gonna evaluate
let's
it.
not make the 1918 virus. What if it leaps out of the Petri dish and causes a new pandemic?
They're evaluating
I think that,
it. They're just going to say, should this be funded? And to the key point here, which I want
you
to
know
keep
what
coming
the
back
dangers
to,
of that are.
is-
You've seen what the dangers of that are.
what are the dangers of which thing?
What, appointing 9 people to say whether this experiment should be done or not, whether it's dangerous or not, on the basis of what?
What does
Well-
the study section decide?
Well, hold on a second. But first of
They do
all,
the same thing.
the public is funding all this, right? So the public is funding all this. So if we can't convince the public that the benefits outweigh the risks, then none of this is going to happen at all. So, and to
Yeah,
this
well,
point, the virology
exactly what I'm
community...
saying.
Let me
is-
jump in just
yeah,
for a second,
sorry.
okay? So right now we've jumped into talking about the Risky Research Review Act.
But it's
if
not a
anybody is-
hypothetical. I mean,
right?
it defines exactly what we're talking about.
Right.
And it says exactly what's going to be done. So we don't have to debate banning. There's no ban. It's just a review. That's what we have
Dr.
right
Gupta
now.
was saying who's going to be doing the reviewing. I think that's the question, right?
People with expertise in virology and national security. I mean, this is a national security issue. These are viruses that are national security risks. This is the part of the definition of them. We are allowing scientists to have
Create
control of our national
security threats.
Yeah, it's absurd. I mean, these are bioweapons agents. What these things are.
And they're publishing online, and therefore the mutations can be picked up by any rogue state or
I
Friends
think we're
of
giving
America or
ourselves
whoever
far
you
too
want.
much credit. We
mean,
can't
free
make
of
these
charge.
things easily, believe me.
So
I wonder if there are ways to bring
it
this...
could be, if I could say, right, it could also be argued, scientists haven't really shown themselves to be very, effective at doing such things over the last 4 years, right? I think that's what's being suggested.
Effective at what things?
Self-governance?
Telling the truth? Sorry. Sorry?
At providing meaningful oversight, was what I was thinking.
Well, that's 4 years. It's been decades. There's no force of law regulation on this research.
Hang on a minute, I thought gain of function
There's no.
research was
It's subjected to, and
highly regulated.
it's subjected to the biosafety community, like the biosafety committees at the individual institutions get to decide, like if the processes are all in place.
Well, how are you going to improve upon that?
They're approved, but what if,
How are you going to improve upon that?
well, so just say if you violate, if you're supposed to do something at BSL 3, and you perform it at BSL 2,
Well, obviously
nothing happens to you.
that is not true. Not
You lose a
true.
grant.
If you do something in BSL 2 that you should do in BSL 3, you could be dismissed.
Let me kind of
Actually, this may vary
reorganize
from
the
different
question a
countries,
little
doesn't
bit.
it?
So let's say that there's a lot of people that believe that there needs to be some kind of independent oversight. This is, there's a bipartisan view on this. There's 2 senators. It's moved out of committee. There's a lot of interest in this. One of the questions we have here, and I think is very relevant, if the U.S. restricts such research or requires oversight, is that not allowing other countries to rush ahead with this research without knowing where it might take us? And I just want to, put communist China kind of front and center here for a moment, because I was just reading, there's a paper in the American Journal of Transplantology, execution by organ procurement. Okay, that has made it into the literature because there's very limited ethical boundary within the research being done there in many areas that we've seen. So the question is, is this something that's reasonable to do, to put restrictions in a situation where there's other places where there are no restrictions at all?
I would say, sorry, can I?
Can I just say that
Let's
this
start with Dr.
absolutely
Kahn.
has to be global? And unfortunately, there appears to have been a reduction in norms with the Biological Weapons Convention, with all of this activity going on. And, we need, the BWC never had a verification protocol. It was, there was a lot of opposition to it, mostly from the United States, for a variety of reasons. But we really need to ensure that global efforts to instate the norms to not create bioweapons. There has to be cooperation, and unfortunately, our current geopolitical situation, there's very little cooperation right now between the U.S. and China and Russia, the countries that have a lot of these research facilities going on. So instituting stricter norms is going to be hard to do at this state and time, but that doesn't mean we shouldn't keep trying. But the U.S. absolutely needs to lead in these efforts. What were you going to say, Simon?
I was just going to say, we built the bioweapons capacity in other countries. I mean, we actually developed bioweapons programs in China, absurdly. I mean, the diffuse proposal was a collaborative effort with China. What are we doing? I mean, it's absurd. So one of the reasons, if you look through the emails and the Slack messages for why there was the cover-up of a potential lab origin, was to maintain relationships with China that would allow us to get access to data. I mean, that looks like one of the key drivers of the people that published the fraudulent proximal origins paper, because they wanted to, tampen down tensions with China.
Yeah. Well,
So-
do you think that was the wrong thing for them to do?
Absolutely.
Why?
Because it used the journals for reasons. The journals.
No, to use the journals, but that particular sentiment of saying, well, we don't want to cause a huge furore over something.
Oh,
probably.
Right. So there's a good question for how should we do it in the future, right? So absolutely, I agree.
I think that.
Why use the journals to do it?
But we've talked about that. That's a problem that's endemic in science right now, is that there are these special privileged relationships, there are cartels, there are clubs, there's tribalism in science. We all know that is a big problem.
I don't think the public knows it. We know it, and we're used to it, but the public has thought peer review is a big deal for them.
I'm sure, and I agree that should be dealt with, but we need to, I think we should park that. But in terms of thinking of this as a cover-up for a lab leak, I think what happened is that they started off thinking, oh, we mustn't let this become a huge political thing. And then once they did that, using their standard means of doing that, then they closed ranks after that. But that does not mean it was a lab leak that got covered up.
We don't know.
But the behavior is very suspicious of people that are innocent. I mean, I mean, the behavior of covering up at that scale is incredibly suspicious of people.
I don't
that
think
did-
so.
And any other thoughts on, creating strict regulation in the U.S., whereas in other places, doors are wide open for anything? CRISPR, babies,
That's for another conversation. I mean,
CRISPR.
the US can take a lead because of its massive influence in life science research in the world. And one thing, Mike, I would like to say now, in case I forget and time is running on, is that we're talking about mainly the US perspective. Europe has vastly underperformed the US in the gain of function discussion. So we're not trying to sort of beat our brows and beat up our American colleagues. Europe has vastly underperformed. Countries with excellent science traditions like Switzerland, Denmark, Sweden, zero conversation. The conversation in Germany has been very limited. France had one discussion 10 years on. So I would like to say, can do better, but you're doing vastly better than anyone else on discussing this. So, well, bravo, that's the first thing. Second thing is that this so-called gain of function, it's a misnomer. It was deliberate rebranding of dangerous research, and it was to cover up this dangerous research. So you come up with this lovely phrase, gain of function, which has been used in genetics for years. And there's huge numbers of experiments done every year, gain of function. It's dual-use research of concern. It's biological warfare, generation of biological warfare by civilians, just like we have the Biological Weapons Convention in the military domain. And of course, in both cases, these products will never be tested because they can't be tested on humans. Either the Fauci viruses will never get injected into humans, and hopefully biological weapons will never be used. So we have the equivalent experiments done by civilians and by the military. We know North Korea and Russia has active programs, and they're unfalsifiable, literally speaking, because ethically you can't put them into people. You can only do it by deliberate injection or by an accident. So we're in exactly the same situation. So when you sort of say, well, why not to make biological weapons if you're a civilian scientist? What's your attitude to a military operation making biological weapons? Do you have the same opinion? Do you say they should go ahead? I would ask you to bear in mind that.
So we're running up towards
Yes,
the
of
end of
course.
our clock, but I get the sense that the four panelists immediately to my right are for this sort of independent oversight of gain of function research, proposals and so forth. And Dr. Gupta, you seem to be not for that. Are you for any kind of regulation in this realm? Or
Well, we
further
already
regulation
have a
of
lot
this?
of regulation, which is, it has to be proportionate. It's just like the lockdown situation. Well, what are we going to do? We're going to do, even if lockdowns or NPIs did stop the spread of the virus, under what circumstances would you use them? So we already have a lot of regulation. You don't do any work outside of, we have a lot of safety rules in place already. We have pathogens that really never harm us. We have to do the work in category 3. We obviously, we err on the side of caution with most things. I think that if the threat is overblown, which I think it hugely is in this particular case, and then you bring in too much regulation, you're basically doing the same thing as we did with lockdowns. You're creating a situation that is unnecessary and will cause damage, will impede research, and will will shift the focus on how to stop a pandemic from happening in the first place, rather than how to mitigate its effects. So I just think the threat is overblown. What we can do we're already regulating this stuff to the extent that it needs. Of course, all this work should be done at a high biosecurity level. Of course it should be. But beyond that, to fret about it and say to stop it altogether, I think is indulging in the same kind of safetyism that lockdowns and NPIs.
So if I hear you correctly, you believe the emphasis shouldn't be on trying to prevent emergence via whatever means, but instead to have very good systems of mitigation in place. Did I read that right?
Yeah.
Okay. So we're about 5 minutes out. I'm going to give each panelist an opportunity for a 1-minute closing statement, starting with Dr. Washburne here.
Thank you. Yeah, it's been really great chatting with you all here, and I hope we can find ways to have more of these conversations, not only for getting to the roots of different views on the origins of COVID and what that means going forward, but also for getting to a point where we can start to creatively think about how we could do science differently, especially in today's day and age where we have so many data systems that could potentially be used as carrots that we can perhaps use to incentivize more transparency from labs or other kind of softer in-between approaches that might help us mitigate the risks without suppressing the research or preventing science from having some life-saving mitigations discovered. So thank you all. It's been really fun.
Thank you. Just two small points. Bryce got me into writing an essay every week on the Biosafety Now website. So if you want to know some thoughts, and if you want to engage in conversation, anything you read on there, with great pleasure. Second thing, and I'll finish with that, is that every week, study sections the world over say no to the majority of grants that are put before them. This is not a limitation of freedom of inquiry at all. It's just the scientific system.
Dr. Nickels.
Oh, okay, good. All right, number one, I got to try to get several of these things in. I don't see an equivalence with the lockdowns. We're really talking about scientists wanting grant money and not being able to get grant money. The emails and Slack chats that emerged that describe the basis for why there was also a desire to tamp down that there was a lab accident was because this would be very bad for business. That's explicitly stated by Ron Fouché in an email, I think it's February 2nd, 2020. He's like, man, this can't, okay, to paraphrase, he didn't say man. If this came from a lab, it's gonna be bad for business. And that has been the common thread that virologists have been using for the last 4.5 years to say, oh, if we do anything to stop this research, it's going to be bad for us, and that means that we need to figure out ways to convince the public that it's for the benefit of them. But the public funds the researchers. The researchers are not the key thing. The public is.
Yes.
All of these laws, when they're discussed, look at the ways people discuss them. They talk about them for the effect on the researcher, not the effect on the public. That's the tell. That's the tell. If somebody is talking about a bill for public safety and they're talking about researchers, that's not the stakeholder there. The bill's about public safety. The Risky Research Review Act is just asking for an independent review board to look at perhaps a few dozen projects, one of which, if it would have been reviewed years ago, might have not caused the pandemic. The other thing is you're close, I think 3 hours away from Reedley, California. And if you are aware of, there was a discovery of a biolab in a warehouse with all these dangerous pathogens, I'm not sure, and a Democratic congressperson is actually trying to pass legislation to prevent that. This is a community issue.
15 seconds.
15? Okay. Right to know, public disclosure. Scientists are hiding where the labs are and what they're working on. The public has a right to know. I think we would agree with that. So that's another thing I'm trying to push for. Public transparency, disclosure of where the labs are, what's in the labs, and just common sense oversight. Thank you.
Dr. Kahn.
Thank you. Well, I would be remiss to not mention the term One Health. One Health is very simply the concept that human, animal, plant, environmental, and ecosystem health are linked. We live in a microbial world. Our bodies are mostly microbial. From the air we breathe, to the food we eat, to the water we drink, we are exposed to microbes all the time. The activities that facilitate a natural spillover, the eating of exotic animals continues. The research that No laboratory is infallible to error. The research pandemic potential pathogens, gain-of-function research continues. So it behooves us to find a way to minimize our risk for future pandemics. We're not going to prevent it, but to minimize it is to do an all hazards, one health approach where we try to address the spillover events through surveillance of the workers, the animals, occupational surveillance, the animals that we're eating, as well as better oversight of the research that could potentially spill over and infect and inadvertently cause a pandemic. So I want to thank everybody for your attention and thank the organizers again for holding this important conference.
And Dr. Gupta to finish off.
The last word. First of all, I just want to say, remember how we've been railing against certainty. So can we please, may I please just entreat you to not think of the lab leak, to not use that same language to talk about the lab leak. I'm sure it is something that should be discussed, and there are lots of good arguments for why that should be the case, and some of the evidence you found. Alex, those should be out there and discussed in a civilized manner. But to talk about the lab leak as if it was certain, as I see many of you doing here, I actually find quite problematic, I'm afraid to say. Then just to answer the last word on Laura's point, I don't think that there is, I think we fear the idea of a pandemic too much. I don't think there are many ways in which we can stop it from happening. I think there are huge opportunity costs to it. All the money that's been spent on trying to hunt down every single virus and try and make sure it's not going to kill us, that money could have been spent on strengthening healthcare systems in the global south. It could have been spent on strengthening healthcare systems everywhere. Let's get these problems into perspective. We have a lot of other issues to worry about.
Well, let's give all our panelists a big round of applause. Thank you.